Welcome back, everyone. I hope everyone enjoyed the breakouts as much as I did. The discussions have been great. And I would like to really reiterate what's been said again and again and again in this meeting. The developmental atlas the HCA itself is a big, ambitious project. And here we're doing all organs that are changing dynamically over time, impacting all aspects of both pediatric and adult disease and regeneration. And it's even more challenging. And this breakout, we really started to address some of the questions that we'd be able to address into the future, and really taking a look into the future where we're not quite ready yet, but on the other hand, we're doing everything in parallel to begin to do what we can do now with these methods and data to have an immediate impact. So we're going to try and start more basic. How can we use single cell genetics, the genetic information at the single cell level to gain insight in this sort of developmental pediatric context? And Gray and Xiao will report back.
Okay, great. Thanks, Dana. I'll share my screen. I've got a couple of slides that I want to go through, if that's okay.
You mean I share the screen?
No, I can do.
Okay.
Okay, can everyone see? It's good. Okay, yeah, so together with Chen, we hosted this breakout session 4 on single cell genetics. And we kind of set up the tone by kind of explaining that, of course, we can measure all of these different features: transcriptome, genome, proteome, epigenome, spatial, lineage, et cetera, at the single cell level. And then we can bring these different modalities back to establish a concept of cell phenotype. And these different cell phenotypes all come together to kind of characterize the tissue or system level phenotype. So I think many of us within the HCA are kind of working on these projects to kind of build up these cell and tissue phenotypes for humans. Also within a given tissue, there are clonalities in which some cells might be different from one another based on their genome. And how can we start leveraging this information to kind of link the genome back to the phenome? This is obvious for things like cancer, but in certain disease and even healthy settings, there might be some within tissue variation that can be harnessed through. Of course, we can analyze between individuals to also see a bit how the genome is linked to the phenotype. And so, with that, and also kind of overviewing a background of organoid technologies and how they might be used to enhance the cell atlas because of their various features, that you can have access to difficult embryonic time points, you can do time course measurements, you can test perturbations, et cetera. So how these can be kind of useful systems to link genotype to phenotype. And so, with this as background, we tried to discuss a bit about how, we can start to get insight into the genes and the pathways and that are linking these two concepts, genotype and phenotype. We did a little bit of discussing about how natural variation can be used here, but I must say that most of the people in our breakout session weren't really involved in these types of projects. Maybe they were in other sessions. We talked, one of the things that we did bring up is that maybe something that could be explored is how mitochondrial heteroplasmy can be kind of read out from the single cell measurements and then linked to the phenotypic states. So as a way to kind of get at more information from the data. And some people in the community are already starting to do this. We talked about what are the implications of natural cell type specific somatic mutations that kind of arise during development and how they might be contributing to different aspects of developmental biology and disease biology. And, we had no real resolution here. We just kind of discussed these topics a bit. And again, we had some discussions about how comparison between species can help illuminate human-specific cell biology. So, comparisons between humans, chimpanzees, and other primates to get at human specificity, but then even deeper comparisons across mammals and invertebrates to see kind of commonalities and distinctions between the species. What I thought was maybe some of the most useful part of the session was trying to discuss practical technologies to link phenotype to genotype. So, one of the things that we discussed, and I think it's on many of our minds, is, what are the best modalities to measure to kind of establish phenotype? And, is it RNA, a tag, histone, DNA, protein, spatial, all of the above? Like, how can we kind of get at this in a good way so that when we do genetic perturbations, what are our readouts? Again, there's no resolution here, but we discussed lots of things. One of the things that was brought up is that whether or not a tag or RNA is a better estimate of cell composition. I'm actually curious what other people might say if there's someone want to comment afterwards. One of the things that we also talked about different methods, crop, perturb, CRISP, other sequencing-based methods for genetic perturbation. There's no consensus on what will be the best tool at the moment. I think one of the also important things that I would like to kind of bring to the attention of HCA is, there's been a lot of work in other model system communities, Drosophila, zebrafish, mouse, established genetic resources to kind of really be linking genotype to phenotype, and maybe less kind of consent teams to establish the same level of genetic resources for the human model system. So maybe ACA can play some role in kind of facilitating these kinds of things. James Briscoe did mention that in the UK, they have set up the HDBR, the Human Developmental Biology Initiative resource. And this is providing tissue to people in UK and internationally to use for genetic studies. And there's a new initiative, the HDBRI, where a group of scientists in the UK are using kind of lineage tracing tools to get at developmental biology lineage in human tissues. And so I think this is a really nice example of kind of setting kind of foundational work to do kind of novel genetic analysis in human tissue. So maybe we can all learn from what they're doing in the UK in this case. And I had tried to generate some discussion about what are some other examples, and maybe if the HCA community have other examples where genetic tool resources are now available, that would be kind of helpful for us. Okay, that's really all I wanted to report back. If you have any questions, you can ask or send us a message.
So yeah, we're going to do, I think, all the breakouts together and then hold a discussion. And I think, you get a gold star and a badge because I think that you are the first person in the history of the HCA that ran under time for their report back. So, gold star for you.
I can do well.
We're moving from, genetics at the single cell level, which we didn't quite discuss in depth, and maybe we could do that in the group session later. A step back, yes, the single cell level and understanding how the context of these things at a very specific cell type, and the HCA has had many examples of where single cell genomics has allowed us to really pinpoint the specific cell type and the role of that cell type, where the genetic mutation of interest plays a role. But now how can we zoom back? At the end of the day, we are the Human Cell Atlas. We're looking at organs and their development and their impact on disease. So how can we actually use this at the clinical level and at the genetic level? So now Helen and Sarah are going to report back on their session and discussing those things.
Perfect. Thank you so much. So I'll do my best to meet the goals of timing and being efficient here. So our focus was the use of development in pediatric cell atlas data to improve diagnosis as well as understanding of disease mechanism in our patients. And Helen and I ran the session together, and I'll be reporting back. So this was our charge to try. We know that at baseline, clinical genetics works to help diagnose childhood genetic disease with genomic technologies. And we were trying to think through how single cell genomic technologies can benefit this field, both by thinking about how healthy tissues develop across age, and then comparing patients with clinical phenotypes to those healthy tissues and samples. So when we went through in our session was a quick discussion, which I'll go through here as well, about how we clinically currently diagnose patients using clinical genetics. And then what we were really taking for the meat of our discussion is what is the current state of single cell data in the study and diagnosis of pediatric disease, and when is the potential and barriers for pediatric single cell data to improve our understanding of disease mechanisms going forward. So currently what is done in clinical genetics is either starting with a gene panel or whole exome sequencing. If a variant is identified in a patient that aligns with a phenotype, and this is something that's in a gene that's been published with a variant that's been found before, it's quite straightforward to move forward working with that data. There are also existing research strategies to take novel variants in known disease genes or novel genes with inherently novel variants in a disease setting to think through whether there are connections, and working with settings like MAVE and Decipher can really help build those connections and ask those questions. But it remains a significant challenge to validate novel sequencing variants in patients with clinical phenotypes. And the way that looks here is an exemplar from inborn errors of immunity, which is another way of saying primary immune deficiency. If you have a patient that you think needs genetic testing, if there's high urgency, you'll choose a fast mechanism. Otherwise, we'll usually use exomes, whether or not they're rapid, depending again on acuity. You may find nothing, and that's one pathway. But if you do, you may either find something known or find something that is not yet known and understood. And we have to go through a lot of validation, ideally, to understand how relevant this is to our patients. And then if we find something important and relevant, report that back to the community. And this takes a long time and a lot of resources and is very important. And what we want to talk about here is moving beyond sequencing to discuss what we can learn about pediatric development and health and dysfunction from single cell technologies. We're merging this with the Human Cell Atlas mission, of course, with a focus on the idea at the end of diagnosing, monitoring, and treating disease. So for the Pediatric Cell Atlas, the new dimension that's been added, of course, is time. And as we move through developmental time, we also see evolution of the systems of the body that we each study in our own labs and clinics. And so thinking through that normal developmental change and then thinking how to connect that to the changes that we see in disease and how to use healthy data to help us understand disease better. So the two main focuses, as I said, were what are the current state of single cell data in the study and diagnosis of pediatric disease, and that was mostly focused on a conversation of limitations and opportunities as they exist. And our real reporting back is going to focus on the potential and barriers for that data type to help our understanding of disease mechanisms going forward. So we had 4 or 5 main thoughts that came out of this really active discussion that was really fun to be a part of. The first was, how are we really going to be able to integrate data across data sets? And again, this is out of generally the people in our group's specialty, but if we're going to, as translational researchers, use this data, it's imperative that it's integrated across data sets and that there's something we can then bring our own data from patients or small studies or large studies of disease and compare it accurately without as much as possible without concern for batch effect. And so one technical recommendation that came forward as a question was, could there be a centralized source of control samples that could be spiked in data collection as we move forward, for example, with GTEx and with the recent CZI, PCA request for submissions, either healthy control cells or a commercial product or a stimulated cell sample, depending on the organ of interest, to try to bring across healthy and disease more ability to control and compare accurately. In addition, there was a lot of discussion around clinical metadata, the need for clear and consistent collection of metadata, ideally that's been shared with other groups doing similar or related work. And by that, we mean organizations and teams that have already thought this through. There's, of course, in the data portal already information that's been included, including things like age, sex, and clinical phenotype, but this could be amplified. And some of this amplification would be helpful around pediatric issues specifically, thinking about, and then of course, like the location of the sample and both in terms of on the patient and where on the earth. But then thinking about with these cataloging groups, how do we think about ancestry? How do we think about what ages we can bin together? How do we think about puberty? And how do we record the clinical metadata so that other groups who will then use the healthy data or use the disease data that's being contributed can most accurately and effectively use the resources and the donations from these families and children that have given to us to study and understand their diseases better. Finally, it was also raised that including clinical sequencing if relevant, that in the setting of pediatric disease, often with early onset disease, there can be a clinical component, and that being able to include that would be very helpful in knowing the relevant variants should they exist. And then trying to, again, optimize our use of data as best as we can when we sample from children who are willing to give us samples in youth, being able to maintain a relationship with those patients and being able to get longitudinal metadata. This patient developed X, this thing happened in patient Y's family. Again, always being very careful to think about the ethics and identifiability of these issues that will give us an opportunity to get more from this data. The next focus was on funding investment needed for optimal infrastructure to foster use. So how can we make the data, again, how do we make sure people know it exists? And when they can get to it, how do we make sure it's easy for them to use? So one of the exemplars that was brought forward was the Allen brain map, and the fact that you can move spatially through that dimension. Here we've added the dimension of age, which they have here as well for the developing brain. There's this sort of visual desire and need to be able to look through the data, sort of in a clinical pathology kind of model of visualization being very important, but then also being able to access the data, find the data, compare to the data that is collected for the healthy samples as well as for disease samples. And there was an emphasis that single cell data is not just transcriptional data, which of course have been said by many people, but the spatial transcriptomics is fascinating, but there's other things that are being collected about these patients, be it exposures, be it ATAC, be it CyTOF, and finding ways to be able to think in this multi-omic strategy be incredibly helpful. So really sort of easing usability and access to the very high quality data that's well annotated that people would like to be able to have access to, recognizing that all three of the things I've just said out loud are very hard to fund. And so funders that are interested in sort of building this tool, recognizing that we all saw it as a real service to both health and research to build this. And then thinking about whether we need to do things in order. Do we need to study health and then disease? Or if we're going to study them in series, how do we do that? And so one strategy that we discussed, are we ready to use single cell as a diagnostic tool? Or do we need to understand disease better? And do we need to understand health before we understand disease, or can we study them in parallel? And so one structure that we talked about was recruiting patients and healthy controls. And this is using the immune system as a lens. Of course, there's many systems. I just happen to be biased, but using this lens, if we can define at a pathway level, which is something that single cells technologies are so excellent at doing, defining the heterogeneity in health and disease, and identify pathways that are dysfunctional and alterations in function of the cells of interest in our respective systems and our respective patients, and then taking that pathway identification, modulating the function of those pathways, either in primary cells or when we're limited in our access to primary cells, creating cells that we can study, recognizing the limitations of that strategy, but sometimes things are necessary to continue to move science and medicine forward. And can we iterate and loop through this by retuning in our respective systems, the systems that are at baseline in our patients not working properly, but so reliant in knowing what health is to look at dysfunction, we need to know what function is. So can we move forward towards pathway-based diagnosis and potential therapeutic targets for patients in that fashion, and then over time, move to the N=1 diagnosis of new patients with undiagnosed genetics or exposure-based disease. And there was a focus in our group on the importance of studying this rare disease as well as common disease while we're learning more about immune health and tissue health broadly. And there was an emphasis that this idea of, and this has been said in many other groups, that health versus disease is not a binary state, and that delineation in a given organ or given person is a real challenge. And that one can argue that going along this model of parallel rather than serial study of health and disease, that we need to include disease cells in the sort of atlas of healthy to really cover the range of possible healthy cells. And then finally, we talked about how analysis takes place and what are the comparisons once we have healthy data that is easily accessible, highly QC, that we can compare our N=1 patients to. And we've been able to catalog our diseases of interest to compare where our patient lives within those two groups. How do we compare across ages and development, comparing rare disease pediatric patients to age-matched controls, and then recognizing in my last 3 seconds that the dedifferentiation state in adult tissue may look a lot like pediatric tissue. So this is very relevant to both adult and pediatric disease. Thank you.
Thanks a lot. So this was a really good breakout and summary. I really think that you touched on some of the most critical things. And, as a computational biologist, I'd like to reiterate that experimental designing the experiment and the metadata correctly, what you collect and what metadata you store, makes the job of the computational biologist afterward and like your bang for a buck and the information you can extract so much more, as well as the importance of querying, which I think has been a theme in this conference or meeting. Again, it's not only the infrastructure and the software, but I think we have a more fundamental question of how do you technically, mathematically, and statistically query, and a great example of, how do you compare disease versus robust definition of normal. Our final report back for this breakout session is regeneration. And I think that regeneration is the best bridge between us and the adult atlas, because it happens at every organ, and there's also regeneration in the adult tissues, and there is regeneration effort in some of the adult atlases. So this is really a bridge that connects the more HCA pediatric and the developmental atlas to the adult atlases. And I'm really interested to hearing, what the report back will say about it. So Jason and Guoji, all yours.
Yeah, thanks for that. Let me share. All right. So this was a session that I led with Guoji Guo of Zhejiang University in China, and Tata from Duke University. We invited him to give a short presentation just based on some of his recent work at really elucidating some of the ways we might use atlases to define regenerative processes in adult humans. So we know that tissue integrity, organ function, and the capacity for endogenous repair all decrease with age. And at the heart of regenerative medicine is really the ambition to regrow, repair, or replace damaged and diseased cells, tissues, and organs. And we spent most of the time highlighting how each of these approaches can be benefited by development in pediatric cell atlases. And some of the main questions which came up, and I'll get to some of our solutions and answers to these questions on the next slide, are what are the genetic and epigenetic differences between developing adult and aged stem cells and even somatic or parenchymal cells? What interactions, pathways, and niches regulate the quiescence and activation of stem and progenitor cells? How do inflammation and other environmental signals, including maternal influences and early developmental perturbations, influence tissue integrity, aging, and repair later in life? How do cells at the heart of it change during disease initiation and early progression? And how do we access samples from those early disease patients? And if diagnosed early, can these changes be reversed? Is there a place for intervention? And what can we learn about the normal developmental trajectory? And how do we leverage that knowledge to develop effective therapeutics? And then I think there was some discussion about how similar are engineered cells and tissues to their endogenous counterparts. And this really speaks to the question of safety of any ultimate cell-based therapy that we might pursue. So we thought about which models could be used to study human development, aging, and regeneration, and spoke briefly about some of the advantages and limitations of the various models. The discussion really centered around the ones that I'll highlight here. So I guess at the heart of it is primary human samples, both pediatric and fetal, adult, and I would include in this trajectory geriatric, because we know that the capacity for regeneration there is decreased. And I think there's something to be learned from comparing younger individuals to older individuals where you have the loss of endogenous repair capacity. So I won't go through the details below, but we talked about organoids and their power there, like has come up in other sessions, their ability to model developmental biology and even the identification of novel intermediate cell types. We spoke briefly about the directed differentiation of iPSCs, both as a way to model development and to test hypotheses that we've identified in developmental biology and the derivation of adult derivatives, but also as a potential to derive autologous cell therapeutics or even heterologous cell therapeutics if we were to safely engineer them to be immunoprotected. And then lastly, we spent quite a bit of time actually talking about animal models, which I think we can all agree are very well controlled. You can perform the experiment that you'd like to control to perform, but they're very contrived. And so we talked about ways that you might optimize animal models, including larger animal models, more complex vertebrate models, including non-human primates, but also this idea of wild mice or kind of environmental exposures in mice to things like Western diets and medicines. So I will just then highlight some of the key discussion points that came up. And I guess we started off the conversation with Tata's presentation, which highlighted that there really are significant differences between mice and humans. He highlighted a couple of populations that we found recently in the human lung that just aren't there in mouse lungs. So are some models better for some tissues, and what would those models look like? So we talked quite a bit about organoids and larger animal models, including non-human primates. We think that multi-omics is going to be very important for the identification of novel subpopulations of cells. It was raised that you might have some transcriptomically very similar cells, but if you look at the proteomics, for instance, their expression and production of various surfactant proteins, you can identify novel subsets of type 2 cells. So how do we integrate proteomic data or epigenetic data with the transcriptional profiles, which have been very informative in recent years? The consensus seems to be that there is a lot to learn about repair processes and subsequently regenerative medicine from studying development, because as I mentioned before, kids are generally pretty regenerative. So what can we learn about that capacity for repair, and how is that capacity for repair lost with age? We've talked quite a bit about sampling tissues from adverse health environments or just different general exposures. So endocrine disruptors, maternal exposures, so on and so forth, to get a better picture of human repair processes and those early causes of disease initiation and progression. And I think has come up in previous sessions, there's a real need to incorporate the metadata that would come along with these and any data that we generate related to these complex human populations. So we talked about different geographic locations or different genetic subpopulations. As has been talked about with others, we definitely recognize the need to incorporate transcriptional profiling data at the geographic level. So we talked quite a bit about spatial transcriptomics and other approaches, and then thinking about trans-tissue mapping to learn about how these kind of universal cell types, endothelial cells, pericytes, macrophages, how are they specialized within tissues, and what can be learned there. We spent some time discussing what development in pediatric atlases cannot cover. In particular, we spoke about fibrosis and some other inflammatory conditions, and what are the limitations there that we should think about. And then we were tasked with thinking about what a message might be to funding agencies. And I think we can all agree that cost is a major burden here, and that we need access to much cheaper single cell technologies. We spoke a little bit about the possibility to deconvolute bulk proteomic data to make some inferences about single cells, but really the ultimate need to share and collaborate across the community, especially as we think about incorporating additional human populations and perturbations. So that's all I had to share, but look forward to discussion.
Okay, so thank you very much. Now, I want to open the session. I think, these three sessions really had a lot of synergy between them. It was a little bit unfortunate that we broke into separate rooms, and this is the time that we can make this synergy happen. And so I'm wondering if we can start with a question, not from the audience, but one of you asking another session a question. Let's open with that. Do none of you have a question? So I will throw a question if no one has. Jason seemed like he was about to ask something. Jason? No, okay. So I guess I will ask a question. I really think that, when we're talking about genetics and regeneration, cell types and cell states are central. And I didn't hear the word, cell state being described. It's genetics that impact the cell state. The cell state can then communicate with the rest of the tissue and have propagating effects to alter physiology at the tissue level. And of course, regeneration is all about switching and translating between cell states. And cell states is a pretty central theme in the human cell atlas and in the single cell world. And I was wondering if anyone here had any sort of something to say about, cell state in the context that we've been talking about, and what's needed, or how they're different, or how they could be used more powerfully in everything that we've been discussing.
I can make one comment there. I don't know exactly if this is what you're going for, but once we have these atlases, what we're trying to do is reconstitute a cell state transition in organoid. For example, with cancer, you can analyze cancer, and then you can create cancer organoids, and then you can control the state transitions by incorporating different cell types in there. And then you can measure it over time and see the emergence of a state that is in a particular version of a cancer. And then once you can show that you kind of make that state transition in vitro, you can then explore the genetics underlying it. So for us, this is kind of how we're playing off atlases. So, not only for cancer, but developmental biology as well. It's similar.
Okay, anyone else want to throw something in there?
I think it's in the context of both rare disease and health, I think on the immune system side, one of the things that's really fascinating to me about primary immune deficiency is you you're basically altering cell state with these two different monogenic disorders. And so you can play with sort of the traditional cell states you'd see in a range of healthy tissues with these individual rare diseases. So for me, that speaks more to sort of the study of rare disease and healthy disease and healthy patients in parallel, and trying to sort of play back and forth between those two.
Yeah, I really think that genetics is very interesting in terms of, like, we've been very gene-centric in biology for a very long time. And, you know, now we're taking a somewhat cell-centric view. And now the idea of connecting genes to cell states, where there's also different new states, but actually we see that actually a lot of genetics alters the composition of cell states, and the composition and frequencies, you know, without even actually sending states themselves are very critical. And I think getting more connectivity and connections and understanding between genes, the individual cell state to populations is going to be a fun time for all of us that will help address all the questions of this panel. We're going to jump back to the audience. It seems that everyone is enamored and interested in all these new modalities. So up until not that long ago, there was, you know, single cell RNA-seq, and everything else was, you know, not high throughput enough to really matter. And now there's ATAC-seq and multi-omics RNA-seq. And, you know, there was questions that were raised earlier about, you know, what defines a cell type better. In the context of genetics, I think that really defines, you have to define what better is to even answer that. I think each one gives you a different aspect, and trying to rank them might be wrong. But people asked about the concordance that we discover in multi-omics versus single cell attack, and I guess versus RNA. And so does anyone want to say anything about the concordance? Okay, I guess I'll have to take that one.
I can jump in and say something about our experience. You know, we've been generating data sets from organoids, let's say, RNA and ATAC separately on different suspensions, and then you try to link them back together by, you know, somehow comparing them. But then now recently we've got RNA and ATAC from different suspensions, and then RNA and ATAC from the same cell, or from the same suspension, and then from the same cell. And now we can start to do kind of quantitative assessments of how they're linked to one another, and also get a bit at the technical details. And we don't have the answer here yet, but yeah, I mean, I think the tools are certainly there right now to kind of get at this.
So we've been actually looking at this for a while, and generally speaking, there's, you know, very high concordance, and you could usually, you know, even without the multi-ohm, like, you can sort of hide the ATAC and say, okay, I'm going to map the RNA to the ATAC state, and vice versa. And actually, I find the degree of concordance actually quite surprising. And while the multi-ohm is slightly inferior in each of these two technologies, it's good enough so that it's also concordant with its singular technologies. And actually in regeneration, at least in vivo of all things, I find, you know, the concordance is the least, where regeneration and the regenerative state in vivo, not in vitro so much, really creates a plasticity where it's a lot harder. There's a lot of RNA states that don't map to individual ATAC states, and you have sort of more promiscuous ATAC states. And I actually think that that's a pretty cool aspect of regeneration. But I think we're still very early in the days of trying to figure out that mapping. Do you want to say something?
Yeah. Maybe I have one more thing I wanted to chime in. That brings, so one thing that we've been struggling with is trying to get the dynamics sorted out between the access, the protein and RNA from these single cell measurements. And I don't know if you've dealt with this, Dana, and how you might, because when you try to map them back from, you know, different cells, you have to map them, you can't see, you map them by what it's most similar to. And so
Well, that's where we're taking a regulatory network perspective, and we have 2-3 methods in the lab that I hope some will come, you know, towards fruition soon, where you can, similar to the concepts behind velocity, where you're using the unspliced to splice, and unspliced comes before splice. If you sort of take a regulatory network perspective, which you obviously have to infer in this case, so, you know, some approximation of it, you sort of understand that, you know, the regulation has to come before the outcome, and that has gotten us some pretty consistent and pretty reliable directionality. And that's been a lot of fun. But all these methods are still, none of them are ready to roll, and none of them are just, methods that take a while to develop so that they're robust, general purpose, and reliable. But we have a few things that we're going to plug out soon. But it's a fun time with all these new toys and technologies. Sarah, you wanted to ask the panel a question.
Yeah, I was interested sort of across our different groups, this idea that understanding normal development of tissues can help us understand adult and pediatric disease. So sort of hearkening to what you'd said in the regenerative piece, Jason, and kind of helping funders and researchers in general see the value of pediatric research beyond pediatric disease and pediatric healthy development, which I think is fascinating. But I think that those sort of common themes between our two breakout sessions may be worth highlighting as well.
Does anyone want to comment anything else?
Yeah, I couldn't agree more. I think there's a lot to be learned, because if we could uncage that potential for regeneration in adults, right? I mean, that would be fantastic. We need to know how to do that safely and in a controlled fashion. And then Bruce pointed out to me that we can learn about fibrosis from pediatric cell atlases, because children and adolescents are usually pretty resistant to developing these fibrosing diseases. So I think that the coin has two sides there, and there's a lot to be learned.
I think also, sorry.
No, Sarah, you go first. You guys are more important than me.
That's definitely not true. But to just finish that small thought, if we can follow people longitudinally, starting from before disease is developed through the development of disease with longitudinal sampling in pediatric healthy who then become pediatric who experience disease, seeing how disease initiates is going to be so fundamental to understanding how disease works, rather than always starting from disease or not disease as cross-sectional time points.
I mean, I'm a really big believer that you really have to have a good grip of what healthy is to even understand disease. What is normal variation versus what is different, and it is a continuum. Disease is a continuum. Like thinking of fibrosis. I mean, I'm an old person at this point. Many of you are young, certainly in the audience. So my lungs are probably full of fibrotic crap, but I'm a generally healthy person, and a younger person would have less. And at some point, that these fibrotic states, which occur much more frequently in a patient that has pulmonary fibrosis or something, just become too much for the lungs to function. But there is a continuum, and I think one of the fun things is that we go side by side. We get a better understanding of healthy. We develop better methods, technological and computational, to sort of see what's different about disease, what is the mechanism, and what we can do about it. And these things do sort of march side by side. So we're going to jump back to the audience a little bit, and I'm going to ask, James Haggood asks, does anyone know of examples where we compare developmental epigenome regulatory network that occurs in regeneration versus development in the same tissue?
While we wait for someone to pitch in, I was just going to endorse the comments you've just made, because there's so much sort of resilience in the system, in terms of many of the major organs will only cross over into symptomatic disease when they're actually quite a long way down a disease track. So that I think that transition from health into disease is happening a lot earlier in a preclinical phase, and we've perhaps got an opportunity to capture some of that by these technologies, and that will be important.
I think one thing that the HCA can do as a central executive office is collect more and more of these examples, so that they can be used generally, by the scientific community to demonstrate to funders, how valuable this approach is. And if I can say something again about regeneration and development, I've looked at both, and particularly in the endoderm. We heard Kat's talk this morning. And actually, development is very regimented. It follows, one direction more or less. There's a pattern to it. It's, you need to you create us from a single cell. Whereas regeneration, there's a lot more plasticity, a lot more unknown, how the injury will occur, what type of injury, and, even thinking about something like the lung, the plasticity of, multiple cell types to multiple other cell types and paths that you just don't even see in regular development and transition from one cell type to another that don't even exactly come from the same origin. So I think regeneration is a lot more plastic and a lot more promiscuous, both in the lung and the gut. I've sort of seen that. We can go to another audience question from Praneet Chaturvedi, and that's, again, how can we improve the cell state detection from multiple data sets? How can we integrate them so that we can sort of really get states that are shared across all the different data sets to get a more generalized concept of cell state? Of course, one of the things that we've never defined as a consortium is cell state, but I guess the critical component is how do we look at, and again, I think this is really a question in the genetics context. Genetics does change us. I'm different than you. And so, once we begin to go from individuals to cohorts of individuals with different genetics and backgrounds and ages, we can begin to sort of factor these things out and get a more pure definition. So does anyone have an idea of, how to do this better and how we should approach this question?
I think there are a lot of tools. I think one of the most active areas of tool development right now is how to integrate data sets. Maybe Dana can also support that. I know her lab's involved in these efforts. So I think compared with a couple of years ago, I think we're equipped with quite a lot of options for trying to integrate across different data sets. So that at least there's, I think, stuff to be dealt with there, but there's some options.
Yeah, I guess, what I could say about, I can say two things about integration. One, integration comes, and I think Fabian Theis recently showed a really beautiful example of this in a work by Malte Luca, that integration is particularly important to be able to identify the rare cell types that are really hard to identify in a single sample where we don't have enough cells. And it is often these rare cell types that are the causes for disease. So we really need to be able to characterize these real cell types. They're a real challenge, and that's where integration is critical. I will say that, integration is hard. Like certainly when we scale up, and if you take one of these autoencoder approaches, you lose the genes. And I'm, a fan of genes. And so I really think that the field should take a switch from let's build this giant integrated 5 million cell UMAP to sufficient statistics that could find these rare states and how to define them and what defines these rare states in a sufficient statistic rather than a single cell manner. And now we'll switch to a question from Gary Bader himself, one of the smartest people in this consortium. How can we take advantage of all this natural human variation that we see in the tissues that we sample to sort of mapping from genotype to phenotype and get a value? So, how, and I'll split that into two, and Gary did that as well. One, how can we do that with the data we have, and how can we try and get a little bit more DNA in order to be able to a lot of the DNA and genetic variation in the data we collect is missing. So is there a way to try and get more DNA captured? As well as, is there anything we can do with the amount of variation that we are able to capture right now?
Yeah, maybe I'll jump in. I was really hoping that we could address that a bit more in our session. I must say we did not come out with a good conclusion. I think one of the things that came out that I thought was interesting was this idea of mitochondrial heterogeneity. So different mitochondrial proportions and genomes within the tissue. I think we can see that maybe from ataxic data, maybe that gets enough of it. The other thing I wanted to say is that in our HCA projects, what we've been doing is trying to, from the same individual for which we sample, is to get fibroblasts to establish iPS cell lines so that we can have a cell that we can then differentiate that tissue that we were studying. But at the same time, we have the genome essentially. It's not the individual somatic genome of the cells, but at least it's the genome. One of the questions I had is, in the HCA, how many projects are actually either saving the sample or sequencing the genome directly from the individuals from which they profile? What proportion of projects are actually generating whole genome sequence and sharing it?
I'm not sure about this. There is a very strong recommendation to save and store and bank the sample more for the imaging modalities. So particularly, I mean, the imaging modalities are finally coming online, and even as they are today, I don't think they're good enough. And so the general recommendation was always, if you're going to take a sample, keep some imaging, and SNAP frozen and FFPE is really great for imaging, so that then we can at least get the spatial component of that same tissue. Of course, this IPS that you're collecting is awesome and amazing, but not it's not easy to do for every type of tissue. There's not enough sample from every type of tissue. I'm not sure how much we can do that, but at the very least, to save a little bit of SNAP frozen tissue for any sample would be incredibly valuable, both for the DNA and also as these the spatial technologies are really now finally exponentially taking off. So Heather from the audience agrees that the continuum, both between disease and normal, but also from early to elderly, is an important axis to be thinking of it, because we continue to age. We don't stop aging when we reach adolescence and adulthood. I wish I stopped aging when I was 18. I see my younger daughters. But the question is, in terms of infrastructure, how can we define quality? There's different qualities, and sometimes it's harder. For example, when I look at things, if you're getting tissue from an aborted fetus somewhere, you're not able to get the same quality of tissue that you're able to get from some type of resection you have access to, a rapid autopsy of, depending how quickly you can get it. You have very different varieties of quality, and certainly when you work with animal models, you can get the best quality. So I think it also depends on the type of tissue you're looking at. There's also more fraileable tissue that you want to get some information on. So how are we going to deal with quality so that we can define it and know at least how good we are in comparison to what's possible, and as we also integrate these tissues together and get hundreds and trying to see what is the real biology, also factor in, okay, this was captured at higher quality, so it's more believable. Does anyone have any thought about that? So let's take a step back. Why would we need quality? I mean, I think any metric, and I think this is an important thing about metrics, is it really depends on the biological question and on the task. And different metrics and different features have to hold depending on your downstream question. I think it's going to be very hard to do something general, a general metric, a general integration. Everything has to be tied to the downstream question and in relation and in context to that. At least that's the main, that's my.
Yeah, I want to, at some point, I think this is a very important issue about the quality of the data, because this is different from the human genome projects. And whereas whenever you have a tissue, if it is not degraded too much, you can always get a genome. And there are different layers of things that will affect quality. And one is like what Dan has mentioned, the time that you collect the sample, and how long you digest the sample. And then the tissue digestion is another layer of variability that will be important. How you collect single nucleus or how you digest the tissue into single cells. And sometimes if you digest too long, the quality is going to be bad, right? I think the only way to solve this problem is that reproduce again, and again. If we have, hundreds of data, then we'll understand, the threshold that which are good quality data, which are bad, right? If every lab is only generating tens of replicates, we won't be able to see it. But if 1,000 labs do the same thing, we will be able to, set the quality eventually.
Anyways, this discussion has been fun, but I really think that you made an important point. The Human Genome Project was an awesome project, and this one is just so much greater in its complexity over the Human Genome Project. And the one thing we have going for us, and I think we've seen that in the past day and a half, and probably will continue seeing that, is that this is a very collaborative community. And I think this is such a challenging project that, you know, really coming together across disciplines in an open manner, sharing our unpublished insights, will be able to get to a meaningful finish line in such a daunting yet valuable and exciting task. So now we're going to get to hear some lightning talks and some short talks, and I'll pass on the baton, and you don't have to hear me speaking anymore in the near future.